Regeneration in amphibia. Regeneration is the process of restoring or replacing damaged or missing cells, tissues, organs, or even the entire body parts. The regeneration occurs in various invertebrates and vertebrate animals. Morgan has distinguished there are two kinds of regeneration: 1, morphallaxis, involving reorganization of the remaining part of body of an animal without much new cell growth, e.g., regeneration of new individuals formed body pieces of Hydra. 2, epimorphosis, the regeneration occurs by formation of blastema. Regeneration in different organisms. Some animals like sponges, sea anemones, corals, annelids and starfish that voluntary fragmentation, and these fragments develop into new individuals. This type of regeneration serves as mode of asexual reproduction. In annelids, if the anterior region is cut off, it can able to develop anterior region like samite before cut off. Organisms like lizards and starfish voluntarily cut off their tail, lizard, and arm, starfish, to escape form predators, and that will grow again. Invertebrates. Eurydial amphibians have an amazing ability to regenerate limbs, tail, eyes, lens, jaws, and even internal organs. Man also shows regeneration, especially liver regeneration, called compensatory regeneration. Regeneration in arthropods is more common during larval stages and ceases in adult stage, where the molting ceases at this point. Regeneration in amphibians. The type of regeneration is seen in eurydial amphibians is epimorphosis. Regeneration is through blastema formation. Eurydial amphibians include salamander and newts. Amphibians have ability to regenerate limbs, tail, eyes, lens, even brain tissues. The steps involved in regeneration: amputated wound heal, ACP form, blastema formation and prolification. Regeneration. Salamanders and newts can take 50 to 60 days to regenerate their limbs. Blastema formation. Blastema is the mass of undifferentiated cells formed from dedifferentiation of well-differentiated tissues and cells. When limb is amputated, the blood clot starts, wound heals, and epidermal cells covers wound surface called wounded epidermis. Then the extracellular matrices, supportive factors like collagen, is breakdowns by proteases enzymes. Breakdown of ECM leads to dedifferentiation of mature cells like bone cells, muscle cells, nerve cells, fibroblast. Then the wounded epidermis thickens to form the apical epidermal CAP, ACP. The blastema accumulates beneath ACP, and ACP releases factor FGF8, and regenerating nerve leads to regeneration. Kragel experiment. Kragel and his colleagues conducted an experiment and proves that blastema holds the memory of its mature form. Blastema holds specificity, and it is a restricted progenitor cell. Muscle cells can arise only form old muscle cells, dermal cells for old dermal cells, and so on. Kragel and colleagues transplant cartilage that can express green fluorescent protein, GFP, to another limb of salamander, then the limb is amputated. Then tracking the GFP cells during regeneration, the regenerated limb is largely consisted of cells derived from transplanted tissues. From this experiment he proves that blastema can regenerate to specific type of cell or tissues same to its previous mature form dead. Nerve-dependent regeneration. Regeneration in amphibian is nerve-dependent. For blastema prolification and regeneration, APC and nerve innervation is important. Without nerve innervation, regeneration does not occur. Singer demonstrated the limited number of regenerating nerve is needed for limb regeneration. Singer experiment. In his experiment he denervated the nerves in limb region. Then after 6 to 7 days the limb is amputated, there is no regeneration occurs. In regeneration process nerves plays an important role other than transmitting signals. It is the production of NAG, newt anterior gradient. It is produced in skewen cells of nerve cells. NAG helps for prolification of blastema and normal limb development. In denervated nerve the NAG is not produced. If the NAG is electroporated into denervated limb it develops normally. Tail regeneration follows the same process like in limb regeneration. Regenerate from blastema formation. Lens and retina regeneration is transdifferentiation. Transdifferentiation means differentiation of one cell type into another without dedifferentiate into stem cell or blastema. The lens only regenerate when the removal of lens, lensectomy, occurs, not regenerate from injury. The ventral iris is highly pigmented, but within the 6 hours of lensectomy iris loses its pigment and start to divide and lens formed. Dorsal iris does not contribute for lens formation. Regeneration of spinal cord also shows transdifferentiation. Pathways. There are various pathways and factors involved in regeneration process. Their works are interlinked to each other. WN signaling pathway, involved in formation of blastema, regulates cell proliferation and also essential for formation of APC, helps in patterning of regenerating limb. Fibroblast growth factor, FGF8, FGF10, FGF2 are the essential factors. Primarily secreted by AEC, regulates cell division and leads to blastema proliferation. Retinoic acid signaling pathway, they are signaling molecule, produced in proximal region of developing limb, reestablish the proximal identity of cells by morphogenetic activity, high concentration in proximal region then distal region, essential for proximal region proliferation, activates MICE1/2 genes which promotes proximal structure, it inhibits the distal genes like HOXA13. Bone morphogenetic protein signaling, essential for development and regeneration of cartilage and bone, crucial for rebuilding the skeletal framework. Sonic hedgehog signaling pathway, morphogen responsible for anterior to posterior structure proliferation, determines the arrangement of thumb to toe finger arrangement, contributes to outgrowth of limb, higher concentration at posterior region. Notch signaling pathway, triggering dedifferentiation of mature cells to blastema. Work alongside with WNTFGF to ensure formation of blastema, prevents premature differentiation, ensure muscle regeneration. PROD1, they are cell receptors binds with NAG and initiate cell interaction and induce proximal distal axis proliferation, mainly focuses on proximal proliferation, homeobox, HOX, genes, HOXA13 and HOXD13 for distal proliferation. These genes are upregulated in distal blastema, guiding the regeneration of fingers, toes, and lower limb segments. Thank you.